Hi there. Okay, let us start. All right, today we'll be like gods creating new life. Okay, my name is Alexander Kell, and you are back to Coffee Breaks with TRANSFAC. So it's a coffee break. Relax, take your cup of coffee or tea. I'm actually drinking tea, and listen about TRANSFAC applications. And today we are going to talk about a very interesting topic. It's artificial design of promoters. And the topic, which is, kind of controversial today, is called, intelligent design. All right. Okay, so let me share my screen and we can start. Let's start from the very beginning. And at the very beginning was the word. Yeah, we believe that the life is created by natural evolution, and we strongly believe in that. But there is this alternative point of view that there is a intelligent design. That basically life was created by some intelligence, which is applies certain creativity and maybe certain rationale to design our life. And actually, in the far future, like Yuval Harari said in his bestseller book, that maybe all life on the Earth will be actually designed by us, by humans, and maybe in some whatever thousands of years from now, people will say, for sure, this is intelligent design of the life, which will be there. But now, when I try to, chat GPT, ask him what is intelligent design of promoters, it draws me this nice picture where you see that it's like kind of not God, but some kind of intelligence creating all these types of life. But still, actually, we're going to talk today about gene regulatory code. Not everything in the life, but actually one of the most important part of it, how these life is regulated, how genes which we have, a lot about genes, how they're switched on and off in different conditions. And without this code, without understanding this code, we definitely cannot design any life, because you have to switch on or switch off a particular gene to produce some proteins in the right place in the right time. And we still have very little knowledge about how to design such things, what is a code. We know a lot about gene code, about how the proteins are encoded inside the DNA by triplet code, and that's a lot was done, and it's very clear, more or less. But how the regulation of those genes is encoded in DNA is very far from being understood. And I have this analogy that actually it's like something, if we have a language of which we don't know the alphabet, we don't know the rules, no grammar, we see it like abracadabra, some letters, we don't know what is written in there. The only thing in this text we know is some little pieces like genes. Them we can understand, but how this Alexander, who is me, what to do, where, when, under which conditions, this is all written in this place, encoded in the sequence around this gene, and we have a very vague idea how to decode it. And so at the end of the talk today, we'll have some ideas how to do that. I don't say that we will know the whole regulatory code, but at least we will know and understand some principles of that. Okay, basically, we had already similar coffee break before a couple of weeks ago, and you can find this previous recording of that talk, of my talk at geneXplain Academy site, and basically I can share this link to you. Maybe you can see it right away. So this is a video, you see it in the chat. So you can, that's where you can see and listen the whole lecture about gene regulation in the code, and how what are the different fields application of it for promoter design. Today, I want to make this part a bit short because we want to have a practical guide how to do it. Like, let's behave like gods and try to create those artificial promoters. Of course, we don't do it with in the test tubes right now, but at least we will design them inside our computers. So, but I will summarize some of the topics which were discussed last time. And basically, we know some bits of gene regulation record, that there is actually two important places, which are not two, but many, but we can classify them basically in two groups. One is called promoter, which is we are talking today, so which is a part of DNA, which is in front of start of transcription of a gene, and it a place where transcription machinery is first organized and then it's running and doing its job. But there are also some other places in genome which are called enhancers or silencers, which may be not necessarily near start of transcription. They can be very far away. And it's generally believed that through the DNA looping and through other mechanisms, those places influencing on the regulation of the gene. Today, we'll talk about both things, about enhancers and about promoters. And also we'll understand that we have to combine all those things together to make our design really, really good. All right, and the main players, I would say, of gene regulation are transcription factors. So these are very tricky proteins, and there are quite a lot of them which are created by evolution. And they are especially in high eukaryotic organisms, and they are actually functioning in the nucleus of the cell. And this is an example of a complex of two proteins, two transcription factors, which is RUNX1 and CBF beta. You see they actually build in complex. And then this complex is binding to DNA in a very specific places called transcription factor binding sites. And they, when they anchored there, they can influence the transcription machinery and regulate the gene expression. Already here, you see that the transcription factors actually tend to interact with each other, and that's very important, actually, understanding towards the understanding the gene regulatory court in general. So how do we do predictions of those sites? I was telling this many times. Again, so we actually, we build those, we call them motifs, which are encoded as positional weight matrices. We collect them in various databases. Our database is called TRANSFAC, where we collect all available information from literature and from other databases. And then once we have such motifs, we can apply them to any sequence in DNA and calculate the score, whether the particular place in DNA is similar to a motif, known motif of that transcription factor. And this way we predict transcription factor binding sites. And actually, generally talking about application of artificial promoters in different fields, I listed them here, and you can actually, I discussed that in details in my previous talk. So you see those areas like tissue and organ engineering, regenerative medicine, gene therapy, and cancer therapy is special, and we'll talk about this today a bit later. Synthetic biology as a general term, but it's a mainly place for playing around with the genes and creating some really crazy structures, and basically in research. And I would like you to answer a little pool of questions, which you will see now on your screen, just to understand what is your main interest. And so you see these are areas of application of artificial promoters, which I mentioned here. So if please mark maybe not more than two, which means please mark those things which are for you are most interesting. And then later, maybe in a few minutes after you do it, we can actually look at the distribution, and then we understand what is your main interest of you guys who are listening this talk. All right, so but we will proceed further and going through these different application areas. Again, I was discussing this in details. It's actually currently really pushing very hard forward. Especially, for instance, it's engineering of different types of tissues and different types of cells from the iPS, from stem cells, and initially, several years, well, many years ago, it was a very simple techniques when one cell was just transferred to another type of cell. But now this whole field is really grown up, and here at the top you see that structures like human chip-on-chip or organ chip-on-chip, where different subtypes of cells which are created from original one iPS cell by many different ways, including actually gene switching on and off genes in a particular situation, either by influencing those cells from external signals, and we'll talk about this later, or by really introducing those artificial structures, artificial promoters, which has to switch on and off a particular set of genes in different conditions. And this way, we are able to create different prototypes of tissues and even organs. So this is a very rapidly growing area. And basically, the synthetic promoter, how they do usually is shown here. So it's something which you have to understand. You see here the structure of a cell where the genes have to be switched on or off in the nucleus of that cell, right? So they have to start transcription. So, and then those transcription factors, either endogenous, so those transcription factors which are, oh, sorry, it's going a bit in different direction. Oops. So those transcription factors which are expressed in those cells, they have to be activated. And the activation of those transcription factors is done through signal transduction cascades. And we will talk about this in practice today. And those signal transduction cascades is actually triggered, activated by some external induction. So, for instance, some growth factors or other types of inducers which would activate those receptors, and they in turn activate the signal transduction cascade, which will activate endogenous transcription factors. They will bind to promoters of those genes and switch them on, and they will start working. Well, a more interesting design when you can actually play a real God, when you put the synthetic transcription factors, so of transcription factors which were taken from another organisms, and then this gives an interesting advantage that those transcription factors, since they are not from this organism, they will bind to specifically designed binding sites and nowhere else. Although that's a questionable problem sometimes, but that's a very nice way of development. And myself, I've been involved in one project which is was very interesting. Like if you really look at the future, where it's called Optogenerapy, where in this project we were designing an implant. So actually a device, a small device which has to be implanted on the skin of a human. Well, we were experimenting that on mice, first of all. And this chamber contains contains actually cells, and those cells are genetically engineered. So they are switched on. In those cells, there is a particular signal transduction cascade, which is switched on by very interesting activation by a light. And because what we did, we actually put there a special receptor, which are known, those receptors are known in some bacteria, and those receptors are able to receive and activated by a red light. So we had actually a little light there in those devices, And those lights were switched on and off by actually Bluetooth. So which means from your mobile phone you can switch on those devices which are implanted under your skin, and then those devices which should be switching on the light, which would trigger a signal cascade in those cells, and will end up in activation of specific genes in those cells. So we will actually activating gene which is interferon beta, which was a therapy against multiple sclerosis. And the promoter of that artificial promoter of that construct was designed by in that project. We also contributed to that design, and that was very interesting project. And you see how far this type of development can go, and that was very exciting to be part of it. Basically, generally in synthetic biology, there are really bizarre things are happening when in different experiments you can actually design different types of cells which have to be communicating to each other through constructs of receptors, signals and receptors, and those genes which are encoding another signal, and then those signals are triggered another receptor. And then first you design such cells and then you put them together, and then those cells are actually self-organized. So they start building some structures which nature has never seen before. So that's really exciting. And you can build even more complex organs. And this goes further, and even quite long ago, this table, this review paper was published in 2016, so it's quite long ago in our very rapidly growing field. But still, you see here a table of those organs which are bioprinted, or there's examples. So there's attempts to do this for blood vessels, for heart, for bone, for liver, for skin, and there are actually quite interesting results of doing it. And for instance, this is a very nice paper where the whole heart was constructed by using these iPS cells, and which were developed in different directions by putting those cells in different conditions, and then those types of cells were put together to build the whole organ. And actually, so far in these fields, those cells are actually created mainly by natural involvement, by putting those cells in particular conditions. And recently, very big breakthrough was that the people realized that material where you put those cells are very important to trigger those cells to develop in a particular direction. So, for instance, before people were attempting to grow cells just on a soft material, and they were never developed in a very far in hard development, for instance. But then they realized when putting them in a hard material, just on a very stable, triggers those cells. So cells have special mechanism of trigger their gene regulation through touching to a different type of material. So far in this type of engineering, the artificial promoters are not well used yet in some fields, in very experimental fields, but not really in a big production. The main reason is that there's still very important issues related to the fact that those promoters are very difficult to design in such a way that they will be not working in the wrong place. So they have to be switched on, only in the place and only in the condition and cell type which we want. But it's very difficult to control, so that's why actually it's still not. We have to still apply our knowledge how to design those promoters in such a way that they will be really working as we like. So for that, you have to know gene regulatory code. But in another field, which is a cancer therapy, here we are more free because our main goal here is to kill the cells rather than to cure them or to create something which will be stable for a long time. Here we simply need to kill the cancer cells, And here the usage of artificial promoters is a very big hope and big direction. So for instance, one interesting area of application of those promoters are the hypoxia. So it's well known that cancer cells and tumors in general, because they are growing very rapidly, and there are places in tumor which actually suffer, in a sense, from hypoxia. So there is not enough oxygen in those places of tumor. Still, the tumor cells are able to survive in those conditions. But actually, we know there's a mechanism of switching on genes, and that's the mechanism which our tumor cells are actually using. They switch on a particular set of genes which are regulated by a transcription factor called HIF1 alpha. And this which activated and stabilized only in hypoxia conditions. And these genes are stabilizing those cells, so they let cells to survive even in this hypoxia condition. But the same mechanism can be used by us to trigger other type of genes which can kill the cells. For that, we need to create such genes and to create such artificial promoters which will be targeted by this HIF1 alpha, and then the gene of my interest will be switched on. What kind of gene? There might be different design of that. One important design here is actually related to so-called CAR T cell. Maybe you know this CAR T cell therapy, which is very famous and very interesting and direction in cancer therapy. When specific T cells are created, also artificially, which have to recognize the tumor and kill it. Right, but again, they can be activated also in the wrong places and start killing some other tissues. To avoid these side effects and also to increase efficiency of those CAR T cells, there's this paper published where authors design a special promoter, which again is a triggering, actually accepting activated HIF1 alpha transcription factors, and then special genes starting actually working there, to which are activating those CAR T cells. Right, so that's a very complex structure, but actually it works. And they showed here in this paper, very recent paper in 2023, that they showed that it's really efficient. More older approach and maybe more practical sometimes is really to put a killer gene on such a promoter. So a gene which would produce some protein which will simply kill the cell. And those kind of experiments were done even very long ago. I think this paper were published in 1991, I think. I don't see here no, 2001. And these authors, they actually created an artificial promoter which contains 6 binding sites for HIF1 alpha transcription factor, just as a tandem repeats, and the rest of the promoter they took from the gene VEGF gene, which is actually activated by HIF1 alpha, and they put under this promoter two genes which just show the effect, and they demonstrated they indeed in the cancer cells, those in hypoxia cells, actually those genes, this construct is actually working. And in this paper, in recent paper, they simply, I think this described here, that's another paper, but in the same direction. What they did, they put under this promoter, they put a gene called, which encodes a protein encoded by actually herpes simplex virus. And this protein is known to be killing everything. It's, if you experience to have herpes disease on your lips or somewhere, so basically you see those destruction of tissues which are done by this protein. This protein is a killer protein, was put under on such a promoter, which is triggered by HIF1 alpha in hypoxia condition. And this they showed in this paper that indeed they're able to kill cancer cells. Indeed, again, from the security point of view, so you have to be sure that this gene will be not switching on in all other types of cells. Maybe other types of cells under hypoxia condition, they will be actually working in the right way. It will be not the tumor cells, but other type of cells, and we don't want basically this gene to be triggered in those types of cells. So to avoid that, we have to understand how to, again, saying the same, how to direct the promoters to be switched on in a very particular situation only. So, and I guess such a simple structure, when you simply take one transcription factor binding site and repeat it many, many times, is too far simple to maintain such conditions. And we'll talk about this today. A little bit, and again, I was talking about that before, but a little bit about application of artificial intelligence in the generating artificial promoters. So you see two kinds of artificial, and that's something, again, to repeat from the very beginning, maybe in 1,000 years from now, when our life forms which will be artificially designed or intelligently designed. The only drawback, perhaps, is probably those forms will be designed not by us, but by artificial intelligence. So this will be a bit tricky and maybe a little bit scary, but okay, why not? We just have to keep those artificial intelligence under a particular control. So, but definitely this is a field now where a lot of efforts is put, and then we want to create such programs, such tools driven by artificial intelligence, which will be able to create artificial promoters. So basically, those promoters will be which will be acting in a particular conditions. And so this paper, for instance, is very interesting. These authors of that paper, they put this idea and they understood very quickly and realized that it's a little bit of different types of AI, which they have to apply here. So, in other areas, usually AI tools are there to recognize something too. For instance, in our telephone, we have a tool which recognizes our faces. In many application of AI tools, they have to be recognizing something to discriminate whatever dogs from cats. So to build a model of recognition. But here you have to go a bit further. First, you have to build such a model, but then you have to apply this model to generate something new. So first, you have to be able to recognize cats and dogs, but then the next level, which may be more difficult, and actually to create a cat, to create something which looks like a cat, right? This is a so-called generative AI models, which are known how to do them, and this is a field where we have to apply them. So, and one important notion here is that this kind of first attempts of creating and recognizing structures in nucleotide sequences, like promoters, like other types of signals in DNA. First of all, of course, many people are looking at the nucleotides themselves. So because learning from the previous experience, when we know that proteins are encoded by nucleotide code, then naturally we apply it also here, saying, okay, the nucleotides of themselves are those letters in our alphabet, and we have to simply decode them in some signals and be able to build such AI. Actually, very quickly, we realized that it's not enough just to talk about single nucleotides. Although this, here you see the table, how those programs of deep learning were applied to recognize actually enhancers of cell-specific type, like, for instance, enhancers of which are driving genes in liver or in brain. And the actually AUC and sensitivity and specificity was pretty good of those tools. But still, to be able to generate something very specific, like a new artificial promoter working in a specific type of cells, that's still a problem. And one breakthrough which was recently done is actually a program called BERT, which is where, just following the success of ChatGPT, when we realized how powerful are those artificial AI tools, which are based on big language models. So actually, and here, indeed, we are talking about some kind of language, as I talked at the very beginning. So we have a language, we see nucleotides, we have the genome, and definitely many features in that genome are encoded in that language. The only problem, we don't know this language. We don't know the rules, the grammar. We don't know, in fact, we don't know the alphabet. So we think the alphabet are nucleotides, but what this BERT is doing, actually the authors of that approach, they understood that the alphabet sort of regulation are the stretches of nucleotides, not individual nucleotides, but they are stretches. So like oligonucleotides or warts, we call them, right? And that's already something very common to what is our general basis of TRANSFAC. So in TRANSFAC for many, many years, we were collecting those warts, right? We were collecting those sites in genome, which are proven experimentally that they play certain roles. So they are targets for transcription factors. They are actually not only factors are not only binding to those sites, but those sites are playing a regulatory role. So indeed, when this notion, when this understanding came to us, then in general, so such approach was built when this program is the AI approach is based on using oligonucleotides as a kind of vocabulary of that regulatory code. And this shown here that this approach shows very good performance. Even very recently in the German newspapers, it was published, another approach which is based on the same idea, which just going a bit further. When again those oligonucleotides, those stretches were recognized as basically most important players of regulation. All right. Okay, there's another paper, very recent paper again, when actually what happens using such approach, these authors were able to generate a number of artificial sequences, and they had a lab nearby. And actually, the topic was, and the direction was to create such promoters, which would be B-cell specific promoters, right? So they were able to have those to design and then to create those promoters, to create those construct in the test tube in reality, and express them in different types of cells, and then measure expression which was predicted by this tool versus the expression which they really observe when they do put those promoters inside the cells. And they see, you see there is a very nice correlation, although you also see that there is big deviations from prediction, which tells us that it's even for such a simple, relatively simple and iterative process, when you can actually design even a better promoter by feedback, so you can learn on those promoters which have predicted better, and then you can learn your rules again. So still, it's a very complicated problem. In addition, it was just B-cell specific promoters. But what about promoters which would be B-cell specific plus they will be switched on only in hypoxia condition, for instance, or under some more complex condition, which you often need the gene to be expressed. And to be able to design such tools, maybe a kind of AIs which are acting as a black box, not enough. And probably here we have to still apply our natural intelligence. And for that, we have to understand in general what are the principles of gene regulation. What are the principles of gene regulatory code? And I will, since we have not much time today to do a lot of philosophy, I want really to go online and show how to design those promoters and how to find out the models of the promoters. Let me simply maybe show one thing, that we generally believe that promoters like our faces, that they have a particular combination of transcription factor binding sites, which you have to learn. But in reality, we know that when we predict all the potential binding sites for transcription factors, then we see something like that. So it's a lot of potential which is encoded here, and that's which is a good actually point, which we understand maybe later, that from one side you can be disappointed that okay, even a small sequence like this, which is a promoter of this gene, when we apply our prediction of transcription factor binding sites from TRANSFAC, we find so many potential binding sites. So you see what you see here, each line here different sites for transcription factors. So the name of motif is on top of them, and they are overlapping. They're like a thick layer of prediction. So on one side, you don't know, after looking at this picture, you don't know which transcription factor is actually regulating my gene. But on another side, it really gives you opportunity. So it means this is a very, rich potential to regulate this gene in a different condition. And this potential, we understand that we have to search for a subset of factor binding sites which would regulate this gene in those conditions we want to understand. And in different conditions, the same gene may be regulated by other subset of factors. And to find this subset of factors, I show that there are AI tools which are maybe recognizing my different emotions on my face, and that the same similar tools have to recognize emotions in a promoter, which means a different subset of binding sites which regulate this promoter in B cells under hypoxia or in different types of very complex conditions. And technically, that happens like that. So if you have if the promoter has several binding sites for several factors, for instance, these three binding sites for three factors, then these three factors has to be activated in the same way. So which means maybe this factor is tissue specific, another factor is only activated under hypoxia, and a third factor maybe some other condition only after the cell is growing, making certain type of connections to another cell. So, and only when all these three conditions are happening together, these three factors are activated. They're binding to their places, to your sites which are located nearby, and then those factors can stabilize this binding by protein-protein interaction, by transcription factor to transcription factor interaction, and only under such condition they will be able to trigger this gene to activate. So, and we recreated a very, I would say, primitive AI tool, which is driven by genetic algorithms. Some people even calling it not AI, but rather machine learning, but that's it's just discussion. It doesn't matter. So basically, it's a very robust machine learning tool, which is able to find such sets of transcription factor binding sites in the promoters of my genes, which are regulating those genes in the same condition. And we'll look today at the one example of a transcription factor RUNX1. Why? Because it's a very, it's one example of such a transcription factor which has a lot of faces. So, and it makes it really difficult to understand what it's actually doing, because there are many factors which are more or less straightforward. For example, you say like, okay, NF-kappa B is always activating transcription factors upon, activation of stimulation of inflammation or immune system, right? Or AP1 is activating genes upon cell growth. So it's a quite straightforward understanding of a transcription factor role. But RUNX1, for instance, and I would say it's rather more exceptions than rules in the world of transcription factors. It may play completely different roles and sometimes even alternative. It can activate the gene in one condition and can repress the gene in a different condition. And to understand how it works and why, again, this will also help us to understand how to create more complex promoters rather than just duplicating one transcription factor binding site, like in case of HIF1 alpha. Of course, it works, but it's too simple. To make it, again, such a promoter more specific, you have to construct this structure more complicated. And on the example of RUNX1, or another name of it is AML1, when it can play different roles in different conditions, we may understand those rules. And to kind of analyze this data, we combine two data sets. Actually, from the same experiment, in one data set, we see actually gene regulation. It's the same. Is it the same paper? Yes, it's in the same paper, in the same work. So they measured RNA-seq. So they did RNA-seq after knockout of RUNX1. So they actually knockouted RUNX1 transcription factor in the tumor cells, and then they measured gene expression after 6 hours after knockout. So there was a conditional knockout. So then they can really take a particular time point when they do this knockout and inactivate this transcription factor. And then they see which genes are upregulated or downregulated after 6 hours, right? Of course, this may be direct targets of this RUNX1. So if there are genes which are downregulated upon knockout, which means they were working when RUNX1 was active, but when you knock out RUNX1, so they switched off. So which means these might be direct targets of RUNX1, or maybe after 6 hours, they could be indirect. So for instance, RUNX1 was activating a certain gene encoding another transcription factor, which is in turn regulating my gene. But then through this chain of events, this gene is affected upon knockout. So this is very complicated to understand how to, for instance, if you want to create such a promoter, which is switched on only in the tumor cells of a particular type upon RUNX1 activation, then we have to know the direct targets of it. And then actually to know a bit more about these direct targets, there's another set of experiments where they did a ChIP-seq, chromatin immunoprecipitation assay, which actually for this RUNX1 transcription factor, and they did it not only after 6 hours, but they did it more rapidly after half an hour, after 1 hour, after 2 hours, upon the inactivation of RUNX1. And with that, and the ChIP-seq actually gives us possibility to find those places in DNA, in genome, where this RUNX1 is really binding. So here we have two sets of data. One set is showing where those genes are binding, those transcription factors are binding, and another RNA-seq is showing which genes are activated or downregulated upon such binding. So by combining these two sets of data, we have a chance to understand the structure of these enhancers and promoters together, which their structure, which would be responsible for be active upon RUNX1 activation. All right, so let me basically go online from this point. And we have here our geneXplain platform, but let me actually start from the very beginning. In in our website, you can log in here. And here, if you don't have your account, you register here and you got the free account. So in the geneXplain platform, you will not get the access to TRANSFAC. For TRANSFAC, you have to get a license, or if you have a license from your university, you can use it right away. But once you have your account, you can log in, and then you get this set of products on our site. And today I want to show you the quick way of analyzing such set of data. So you see we have RNA-seq data and we have ChIP-seq data. For that, to analyze them completely in a pipeline, we have this Genome Enhancer pipeline. You just click here. It gives you a simple interface how to put those sets of data inside this tool. I will show you how to do this. So basically what you do, you create your new project, or, well, actually I have already created a project, which is, respectively, this Intelligent Design of Promoter Practical Guide. So this project, and we have this drop box in the middle of your screen where you want to drop your files. And I will open this gene expression omnibus entries. Let me find them there or here. So first, let's have a look at this entry, which is RNA-seq data. And you see here, there is a description of that data, and there are knockouts. So some, they did, several replicas, technical replicas. So they have a cell line, they did knockout, they did triplicates, I think, and they have a, wild type, and they have knockout for RNA-seq. X1, they also have another knockouts, but for study today, we are interested just on RNA-X1 knockout. So, and they have this all data already here. And basically, let me see, I think that's an Excel file. Let me download it. Right, so let me open it. I think I have to move this away. All right, so this is the Excel file. It looks like that. So where you have, we have here the sort of how many, 23,000 genes. And this is a gene symbol. And we have here all the conditions. They are encoding those. So you have to look here to see which one is corresponding to which one. You have to look in each of these to see actually the code. Yeah, here you see the code to understand which one corresponding to which one. But you don't need to learn it right away. I have to have written it before. I went through all this information. But important is we have this table, and these numbers are the read counts. So when you do RNA-seq, and then basically for each gene you count how many the number of read counts corresponding to expression of that gene. So you see some genes are very low expressed, very few read counts, and some of them are highly expressed. So this may be constitutive factors. So basically expression of a gene can be always very low or always very high. But for us important, what happened with this gene after knockout? Some of those conditions are actually coming from knockout, and I will see actually 1, 7, and 13, and 19 are the wild type, and the rest is knockout. Good. So what we do now, we actually basically upload this table. So we add this file here, and we go here to downloads, here this table. So you see it's uploaded, and we would go for import. And here we have to define what type of data we are working with. So basically this is transcriptomics. And as you can see, there are other 5 types of omics data which we can put in the system. So, and then the system will analyze using all these type of omics data together. So it will actually combine them together. And in our run now, we will use transcriptomics, and then the second set of data are coming from epigenomics. All right, so but for transcriptomics, it's very easy. The only thing we have to actually select is a correct column for ID. And as you can see, the first is entrance ID, and the symbol, gene symbol is here. I would actually better choose gene symbol as a symbol, as a ID for a gene, and then import this. So now we have this importing. So it may take some time, but in the meantime, we go now to the ChIP-seq data. Here we have ChIP-seq data is more tricky. So here you see they already prepared as a peak, a table with peaks. So sometimes this data, if you are dealing with raw data and you have just FASTQ files, then you have to align and create those peaks by yourself. And that's also possible to do here if you, in the system, in Genome Enhancer, if we put here not just ready peaks, but FASTQ files, then the system would first align them to genome and then do the peak coding. But here we have the peaks. And actually, instead of loading them, what we can do, we can directly copy the link address. So just to show you that uploading can be done also, not necessarily by uploading it to your computer and then downloading to your computer and then uploading to the system. You can upload it directly from URL. So actually, you put this URL here, and you click upload. And as you can see, it's also can be zipped. So the system would recognize, okay, this is zip data. It will automatically unzip it. So we will also click on import. And here, actually, it's epigenomics data. And okay, let's maybe do one thing, because we don't know how these tables look like. And we don't know which column to use as accession numbers and anything. We haven't seen it before. So the trick here is to actually tell the system just load as it is, and then we'll see how to convert this table and to which column to use as ID. So for that, we simply look what type of format we have to choose. It's probably tabular format. And then we'll generate unique ID. So it's already ticked by default, by just have to check it. So it will actually take the table as it is and generate just a unique ID by itself. And then later we will understand how to deal with such a table. So, and then we importing it's here. So we have to wait a bit because what it will doing, it's now actually unzipping all these peaks and putting them as a table. So the table of genes is still importing. This one is done, but what we can do is just go next, and to see what we can do. All right, so as you can see, what it did actually, it imported the peaks like a table. And indeed, it was right not to take it right away. You see, this table had a lot of things in the first lines, so it may be distorting. But from the line 30, the real data starting, it doesn't actually name the columns, unfortunately, but we can guess. This is a chromosome 1, and this is a from. This is 2. And somewhere should be also, well, in case of ChIP-seq, there is no information about which strand it is, because ChIP-seq has no orientation. So it's just from 2 and the chromosome. That's the information which we need to analyze this data. All right, so, okay, so, and the expression table is here. Let me see, right? That's the table which was loaded. And as it is, so we can see these 23,000 genes were loaded. So we have the transcription RNA-seq expression and the ChIP-seq expression, ChIP-seq information. All right. So one point is that this ChIP-seq information, this is just a table, but we really need to map it to the genome to know that these are really places in genome to give to the system this information. And we have to do a couple of transformation here using the tools which are behind the scene. So you see now we are using Genome Enhancer, which allows us to do everything automatically. But in case something has to be done with the data before you start the analysis, then you have to go back to the platform itself. The platform is actually the platform, the software platform, which is staying behind all this Genome Enhancer tool. So now we have here additional tabs, which allows us to select some sets of analysis tools. And the tool which we want to use now is basically a conversion. So we want to convert this table into a track. Track is actually another type of file which just shows positions in genome. So and we have here in data manipulation, convert a table to track. This is a tool. We double click on it, so it opens here. And then we take this table. All right. All right. So actually what we have to do, okay, let's see if we have to know it's from 20. It's from line 30, and the first column is chromosome, and column 2 from 2. Okay, so we know which columns to use. So which column? This column is a chromosome actually. This column is from, and this column is 2. And the genome we want to map, and for that we have to know. Actually, it's a latest build, which is HG38, which we are using, and we have to show to the system this is a build which we want to use. All right. So and then this is a peak. And then let's run this. Let's see. All right. So it first it gives that first lines are not really relevant because they were empty, but others it will convert. All right. So it now put this actually peaks on the genome, and to see a bit more what happens on genome, what we can do, we can put here the gene information, for instance, and so then we see a bit more where those peaks are located. So let's wait a bit when the gene information is updated. So this is the first chromosome. We can look them also on the other types of chromosomes. All right. Okay, so but at least we can now see those these peaks. We can also open them as a table, and we see those peaks, right? All right, so it's now opening. And one thing we have to do is to mark this as a type of information we are dealing with. All right, so this is actually we have to set an omics type, which is now epigenomics. So ChIP-seq data is epigenomics. So we set it as a omics. You see it get this little E in front of it. So these tracks are epigenomics data. And all right. So what we now see, let me jump here. We see, for instance, these are peaks, all right? So there are maybe a number of them. These are 3 peaks. And this is a gene. And for instance, we see that those peaks are located inside this gene, and this peak is inside this gene. So we see the kind of topology of where those peaks of binding of this factor are happening. And one thing you have to understand that it's not always in a 5' or 3' area of a gene where the transcription factor is binding, but sometimes it's inside the gene. So, and we know that introns of the genes, they often contains sort of enhancers. So then when we will build our artificial promoter, we have to actually deal with that. So we have to build a promoter and enhancer together. All right, and then, so now we have all the data together, so we can return to Genome Enhancer tool. And what we do now, we actually drag and drop this information in this great table and prepare our tool to work. And we have to specify here, this is, for instance, now a wild type. And this is a, let it call RunX1 knockout. All right. And then what we do, we actually will say that this ChIP-seq was done in the active. So it's actually, this, we have to read it, but I read it before. This ChIP-seq was done before knockout. So, so we took these peaks which were before knockout, right? So then we put it in the wild type, those peaks. And then we take those RNA-seq data, which are 1, 7, 13, and 17, and 19. All right, let's just drag and drop here. This is our wild type. And the rest is knockouts. So these are RNA-seq data from knockouts. We put them all here. So, and by distributing this into these boxes, we just tell to the system what type of data it is, and also which data to take together. So which will take these peaks and RNA-seq data. So which means it will first find upregulated genes, and then we'll check whether we see the peaks of binding inside those genes or in the vicinity of those genes. And then from here, it will take those genes which are not changed or downregulated upon knockout. And then it will start comparing. So then we press next. And here, what we have to do, we have to select a disease. So we are interested here in a general condition, in neoplasm. So for instance, we want to build a promoter which will be activated upon RNX1 activation. And that's actually important, for instance, in metastasis of several types of tumors, or in some special types of tumors, like for instance, leukemia. So that's why this transcription factor is also known as AML1. So, and that's in those particular type of tumors, we want to be able to create a promoter which will be activated in those types of cells. So, but to build this condition, we can either select, for example, leukemia, leukemia, for instance, and we would compare wild type to knockout, and that's it. And then we start analysis. Well, it will take basically several hours, so we don't have time now to wait when it's ready. So you'll see it in the results. But of course, I did run this before, and we can have a look at this. Well, we will maybe better pause this we terminated. Okay, so because not to compete with our speed of the server, but let me show you the results which we already obtained, and we also return to screenshots in a moment, so that maybe it will be easier for you to grasp the results. So here the results which we obtained when we were running. So we have to basically go to the platform, and let me show you those results. So this was done in this project, and so there are a number of big number of results. So it generated these results, which we will see in a moment. So for us, for the promoter design, the only important part now is what is a model of enhancer, actually, in this case, because as you know, this is here we analyzed RNA-seq together with ChIP-seq data. So which means we are looking not only in the promoter of those genes, but we are looking also in those places around the genes which are upregulated, which are target for RANX1. So which are playing as an enhancer for activating those genes. This is a hypothesis. So, and this tool is actually, while it's working and doing many things, but for us it's important now to see this model. This is a model of enhancer which this tool is building using this genetic algorithm which I showed you before. So it gives out of all possible motifs which are available in TRANSFAC, it selected this how many 11 motifs which are together are organized in the clusters. And you see here the for instance this AML motif is a part of those of that model. So which means the AML sites are there in the enhancers because the RANX is RANX-AML1 are binding to those enhancers, but those enhancers has to get additional motifs around those sites to make them really specific and to mark those genes which are upregulated. All right. So, and to see it in more details, we can even click here to see where exactly those enhancers are located and what is the structure of enhancers. We, as you can see, we can sort by this CMA score, which is how good is the enhancer in terms of cluster of binding sites. And indeed, you see this is an area of a ChIP-seq peak. But inside this area, the algorithm found for us a cluster of binding sites of those which it found, which kind of mark the enhancer area. All right. So with that, we have the structure enhancer. So now we are happy. We know how the enhancer look like, but we should be able to apply this to build new promoters. And there are additional tools which are able to do that. And let me simply show to you some experiments I did here with designing new promoters for those enhancers, well, which contains those enhancer and promoters together. So these are my experiments. So what I did, I took actually an enhancer which is located in EYA2 gene. Why? Because actually this is a gene, very important gene which we know involved in metastasis, and this is actually one of the most upregulated gene, downregulated gene upon inhibition of RANX1. So it's a direct target of RANX1. And it has actually 3 peaks inside that gene for RANX1 ChIP-seq, peaks. So that's a definitely a very good target. So what I did, why not taking the enhancer of that gene, take the promoter of that gene, and create a construct, artificial construct, which would contain promoter and enhancer together, like here. So like we take an enhancer of that gene and put in front of it the promoter of that gene. And then we'll see how, first of all, how this construct would look like from a point of view of that model. So our tool can be used to apply this model to any sequence of our interest, so to this artificial construct. So we'll see where actually on this score level, this newly constructed enhancer is laying. And the second, actually, we can do things with it. So for instance, if we look at this enhancer more in details, so I have it here, just to show you how it looked like. All right, so this is a part of genome where this gene is located. And as you can see, we have actually three peaks of RNA, well, even more located on 3' end, but this one are closest to the start of transcription, which we believe they are involved as enhancers of that gene. And so if we look at more details of those enhancers, so for instance, take the number 3. These are binding sites for those motifs which we found as important. And you see this is RUNX1 and AML. Motifs are here, but also motifs for other factors. They are shown here. So basically, by looking at each motif, we can see that those motifs have a certain score. So what we can do, for instance, here to improve, we want to improve this promoter to make it even working more strongly. We can maybe improve it by increasing the score of those sites. For instance, changing nucleotides in this sequence that they fit better to this consensus. And with making this, we can improve these binding sites. We can also do some deletions. For instance, you see these two binding sites are kind of standing a little bit away from the core of the enhancer. So if we delete this part, for instance, we bring them closer, we may improve the cooperation of this factor with these factors, and this may lead to improving the activity of that enhancer, right? So we can do many things. That's exactly what I did here. So I here, for instance, I modified binding sites for AML1. I actually duplicated it, so made a tandem repeat of this AML site. Here I duplicated AML and also glucocorticoid receptor. And I also put this deletion, which I was talking about. So this is my the best candidate for best promoter. And here I also improved the glucocorticoid receptor site. I actually put some mutations in that site to make this site closer to consensus, hoping that such a design will make our promoters even better. So I made different attempts here, and we have a tool of which here in our system. So what we do here, we click here, and we say row, and we put here this file and just name it, like CMA, for instance, FASTA. All right. So it recognizes this is a FASTA format. And so now we have this FASTA file here in the system. It's shown here. So basically, these are all my artificial promoters, which I was designing. For instance, this one is my my favorite promoter. So this is a promoter. All right. So what we want to do, we want to apply, the tools to recognize whether this promoter is good from a point of view of that model which we designed. All right. So what we do now, first we apply. So we analyze this promoter using site search. So here we have the site analysis, and we would actually simply site search on track. Site search on track, right? And so we take this FASTA here. All right, so we have to select the custom, and this is a FASTA source, right? This is the right source, and here we will have to apply whole set of transcription factor library, which we have in TRANSFAC, and we have them here in different profiles. This is a whole library of our motifs, of our positional weight matrices. So we will take this one and apply here. So we are ready. So basically then we run it. So what it's doing now, we take these FASTA files, and actually the same way you can do with any FASTA file. If you create your promoter, or if you take this FASTA file from your other organisms or any FASTA files, and you see it found so many sites that it even doesn't show them individually, just show the profile of sites as such a curve. But maybe you can see in some places, yeah, you see such a dense prediction of binding sites as I showed you before. So now we have to apply our model from this promoter design, to select those motifs out of the predicted ones, which are defining this promoter. So for that, we actually, what we do, we will apply such things like this. By the way, if you don't know which tool to apply, what you can do is simply look at those results which we have already done, for instance, in our examples, which we also provide, and look how this was done. So in info box, if you click on the results which were obtained before, in the info box, you see all the parameters, how these results was created. So with that, you see the history. And actually, you can reproduce it by clicking here, result of analysis. And it opens this tool exactly as it was, with all the settings of the parameters, which was done to do this, to get these results. So what we will now do, instead of the promoters, instead of these promoter sites which were used here, we will put here the sites for which were just calculated. Here they are. And we apply this model of that promoter and enhancer together, the model which we created by running this Genome Enhancer. So here we are. Now it will take those sites which were predicted here in our FASTA and apply this model from this promoter in design which we were running for RunX1 enhancers. And now it's created this. So, and it calculates actually the score. And actually, I applied here a model which is model of a promoter. So as remember, our artificial construct was containing the promoter, which was on the right side, and enhancer, which was on the left side. So from a perspective of promoter, so if we sort it by the score, you see those ones which are we do in our modifications get very little score. Actually, promoter is not good. But this promoter is actually all of them are quite good, so they have very high score. But we still don't know what is going on with the enhancer in this place. So for that, we have to do the same, but apply another model, not this promoter model, but an enhancer model. An enhancer model was here in our Genome Enhancer analysis, and this was this model. We can have a look again at this model. You see, this is a model of enhancers which were constructed, so we will take it. We'll put it here, and we will give a different name. All right, and run it again. All right, so you see how important is this feeling that now we are designing new promoters, we are kind of designing new life, and then we'll see how these promoters can behave. Of course, the final result we can only see when we create such promoters, design them, and really, design them in test tube and test it. All right, so, and as you can see now, actually, if we compare, this was the original score of the enhancer and promoter together. No, this is just enhancer, right? And the score was 10.26. This is the original enhancer of this gene. But what we did, you see, remember, we repeated some sites, we did some modifications, and the maximum score which we obtained was from this one, where we actually deleted this region, as you said, and we duplicated AML1 and also duplicated glucocorticoid receptor. But you remember, we were actually doing glucocorticoid receptor modification, but the score of that is lower than this one. That was a surprise for me. And then when I looked more carefully, I found out that actually by modifying this glucocorticoid site, you can look even more closer at this site. I was modifying a site for glucocorticoid receptor, this site. I made it this site closer to the consensus, but in the same time, when I was doing it, I was accidentally touching this STAT1 site, which was overlapping with it. So I made GR better, but I made STAT1 much lower, and this created, a lower score when I choose those original glucocorticoid receptor sites. And that's important to understand that those sites sometimes overlapping, and if they are not so good, they are not so close to consensus, maybe that's for a purpose. So in the natural design of that promoter, those sites were created in such a way that they were enough to bind GR glucocorticoid receptor to this site, but they also not avoiding a binding of STAT1 to the same site. And if you make GR more strong, you will lose STAT, and this will destroy the whole regulation. So this actually shows us this regulation is much more complex than just repeating the same binding site many times and then hoping this will drive your promoter in the right way. All right, so today I was pretty long, so sorry for that. So we are talking here more than 1 hour. I would say, so I still have a lot of material to show, but maybe we'll do it next time. So put your, put this website of geneXplain Academy somewhere in your book market. And here we actually publish all the coffee breaks in the future. So you see, for instance, this was about today, and in 2 weeks, there will be another coffee breaks, which is devoted to CRISPR synthetic lethality. So it's a very interesting topic. You can also here see all the past sessions. So for instance, this is the one which was done before on artificial promoters. So you can also listen that presentation, where one part which I didn't touch here but was present there. For instance, how to design promoters, more smarter promoters for this HIF1 alpha. I made some attempts also there and shown in this talk. All right, so thank you for that. And if you have your questions right away, so I still have a couple of minutes to answer your questions. All right, so here we see the results of the pool. I hope you see it in on your computer as well. Right, so this is different from what we've seen last time, actually, but I'm happy that my guess was correct. So most of you are interested in actually cancer therapy, and of course, gene therapy related to that. Synthetic biology is clear, and in general, biomedical research. And we were talking today about how to use artificial promoters in cancer therapy. Thanks a lot. So far I don't see any questions, but if you have your questions, don't hesitate to send us by email or just put it in this form on gene regulation. And by the way, here it automatically shows you also promotion. I didn't intend it to do that, but okay. If you're interested in licensing TRANSFAC, here's a very good offer for the Black Friday, which is end of November. So you actually can save up to 1,000 euro. So don't miss your chance. Anyway, thanks a lot and see you next time. Goodbye.